Welcome to the channel. In the previous video, we looked at how cyclins and CDKs drive the cell through the G1 and S phases. Now let us look at what will happen to a cell which is progressing through the G1 phase but has damaged DNA. Such a cell wouldn't want to enter the S phase and replicate the DNA that is damaged. So how would the cell pause the cell cycle in such a situation? So here we have the DNA with a double-stranded damage which is recognized and bound by a complex of proteins. One component of this complex is the ATM kinase, which acts on a checkpoint kinase called CHK2 by phosphorylating it and activating it. Similarly, DNA with single-stranded damage is recognized and bound by a complex of proteins consisting of ATR kinase, which acts on the checkpoint kinase CHK1 by phosphorylating and activating it. The activated CHK2 or CHK1 will then act on a protein called CDC25A phosphatase by phosphorylating it and inhibiting it. So what is the consequence of CDC25A being inhibited? To understand this, we need to understand its role in cell cycle regulation. For the cell to progress through the G1 phase and enter the S phase, cyclin E-CDK2 is required. But this CDK2 has two inhibitory sites that are phosphorylated, causing the CDK2 to be inactive. To activate this CDK2, CDC25A, which is a phosphatase, will remove these inhibitory phosphates, causing the CDK2 to be active and allowing the cell cycle to progress through the G1 phase and enter the S phase. So when activated CHK1 or CHK2 phosphorylates and inhibits CDC25A, the inhibitory phosphase on CDK2 will not be removed, causing CDK2 to be inactive and the cell cycle to pause so that the cell has the chance to repair the DNA if possible. Another thing that happens with double-stranded DNA damage during the G1 phase is that the ATM protein kinase and the CHK2 checkpoint kinase that it activates will both phosphorylate a protein called P53. Phosphorylation stabilizes P53 and prevents it from being easily degraded, thereby causing the levels of P53 to rise. P53 is involved in activating the transcription of the P21 gene, leading to the synthesis of P21. P21 is a CDK inhibitor which binds to the cyclin E-CDK2 complex, causing the CDK2 to be inactivated and the cell cycle to pause at the G1 phase, giving time for the cell to repair the DNA damage if possible. If the DNA is repaired, the cell will progress through the G1 phase, then enter the S phase, duplicate its DNA, and then finally enter the G2 phase. Now let us look at what happens during the G2 phase. During the G2 phase, the cell has an abundance of cyclin B CDK1 complexes required for G2M transition. But as soon as these complexes are formed, a kinase protein called V1 kinase will phosphorylate at two inhibitory sites on CDK1, thereby inactivating it. When the cell is ready to enter the M phase, CDC25C phosphatase will remove these phosphate groups so that CDK1 can be activated and the cell can enter the M phase. Now what if the DNA of the cell during the G2 phase is damaged, or if the DNA was not completely replicated during the S phase? This cell wouldn't want to enter the M phase and undergo cell division with this undamaged DNA. So how would the cell apply brakes on the cell cycle in this case? Just as we saw previously, ATM and ATR kinases will phosphorylate and activate CHK2 and CHK1 checkpoint kinases respectively. These will then phosphorylate and inhibit CDC25C this time. So with CDC25C being inactivated, CDK1 will remain inactive, resulting in G2 arrest. And that's it for this video. In the next video, we will look at how the M phase is regulated. If you found this video helpful, do consider liking the video and subscribing to the channel for more such videos. Thank you.